Hello, everyone. Welcome to this program entitled Aging, an Example of Vulnerability, the Impact on Environmental Factors. This program is proposed to master and PhD students by EUR-Live Graduate School in UPEC. My name is Geneviève Dorumeau, and I am professor of physiology at Université of Paris-Est Créteil. And I am leading a team dedicated to senescence in EMRB. I have a pleasure to be here today with Isabelle Coll, who is professor of chemistry at UPEC and researcher in environmental science at LISA, a laboratory dedicated to the study of atmospheres, and also Robert Barouki, who is professor of biochemistry at the University of Paris-Descartes School of Medicine and director of the INSERM Unit 1124. So, what are our educational goals today? The aim is to introduce you to the cellular senescence and to establish a link between cellular senescence and aging. We would like also to present the conditions of premature aging and focus on the role of the adipose tissue. And finally, we want to explain how air pollution may impact aging. So during this program, we are going to understand and discuss the cellular senescence and aging, scientific and societal challenges of air quality, and have some new aspects on the exposome, which is a useful concept. So the first topic of the discussion will be the link between cellular senescence and aging. And in order to open the discussion, I will give you some examples of premature aging. So first, what is cellular senescence? Cellular senescence is now established as a common denominator to age-associated diseases. A cell that is represented as this yellow circle may undergo senescence process pending the fate of the cell. In a quiescent organ, DNA damage or oxidative stress may lead to premature senescence. These cells are not going to stop their division because the organ is quiescent, but they are going to accumulate in the organ and to participate to the loss of tissue regeneration, participate to tissue remodeling, and mainly release a special secreton, which name is SASP for a senescence-associated secreton phenotype, which is going to release mediators associating pro-inflammatory cytokines, but also pro-fibrotic cytokines. The other way of cells to enter senescence is the replicative senescence. These are cells which are in non-quiescent organs, and the increased cell turnover leads to the decrease of the telomere length, which are a very important structure that are protecting the chromosomes. Whilst these telomere lengths decrease, the turnover of the cells is decreasing, and these cells stop replicating. So the link between cellular senescence and premature aging is illustrated here. On the left side, you can see that the cells which have a fate of which is normal are associated with healthy aging. Here we come with a breakthrough concept that abnormal fate of the cells, and especially if you consider cellular senescence, may overarch many pathologies, whatever cancer, cardiovascular disorders, infective diseases, and other pro-inflammatory diseases, but are linked finally to a modification and an exit of the trajectory of healthy stages of the cells. When the cells are accumulating, as is displayed on the upper right panel of the slide, we have progressively the impairment of the organ which at the end contributes to the disease. At the opposite, when the cells stay healthy, you have an organ which is behaving well, and you have healthy aging. So this is something which is underlying the concept that premature aging may overarch many diseases. And I am going here to give you two examples of premature aging on the next slide. These examples are linked to the chronic pulmonary disease, such as chronic obstructive pulmonary disease, which are now well-established model of premature aging, where the lung are progressively undergoing senescence in different cell type, including the epithelial cells of the bronchia. And we can reproduce in preclinical model in mice the disease by inducing senescence in the lung cells. And this is quite interesting. Another model of premature aging, which links our topic today with the adipose tissue, is the obesity. Now, obesity is becoming also an established model of premature aging, and we will see that in the next presentation, that adipose tissue senescence is associated with obesity, but also is associated with aging. And this, in the context of obesity, you have to understand that the adipose tissue is one of the most important organs of the organism in terms of weight, at least. And this represents a serious burden, since the visceral adipose tissue, especially, is going to release many mediators that may lead to what is called an obesity disease, which relates several abnormality in different organs, leading to cardiovascular disease, renal dysfunction, but also hepatic disorders, together with metabolic disorders such as diabetes mellitus. So this is the introduction to the fact that the link between cellular senescence and aging is an important topic when we want to think about life trajectories. And instead of focusing on a single organ disease, it's important to consider the crosstalk between the different organs and evaluate the load of cellular senescence in order to prevent the further deterioration of these organs.
Geneviève, may I ask a question, please? I mean, since you have been talking about the obesity, what do we know about the role of adipose tissue in the process of premature aging?
Well, the adipose tissue is a very important organ, as I mentioned, and as you will see in the next presentation, there is the cellular senescence occurs very early with the start of the high-fat diet, for instance, in preclinical model. So now obesity is really recognized as a premature aging disease, and all the disorders which are associated with obesity are not linked only to the increase of the fat mass, are not only increased with the metabolic disorders which are related to the insulin resistance, but are related to the senescence secretum of this visceral adipose tissue, which disseminates toxic messages and danger messages to different organs. But I think that you will get more information in the presentation by Zenab Mesdari, who was a PhD in our lab and who did her work on the cellular senescence in the context of obesity. So let's have a look at her presentation.
I would like to thank the conference organizers for this opportunity. So my work that was done under the supervision of Professor de Rumeau in the Mondor Institute of Biomedical Research in Créteil is about the key role of adiposity or obesity in premature senescence. So this study is in line with recent studies published by our team and others, demonstrating that in the context of obesity, as well as aging, visceral adipose tissue has a key role in promoting metabolic dysfunction and cardiac dysfunction with remodeling through profibrotic and pro-inflammatory secretome, and that these secreted factors are coincident with the accumulation of senescent cells in adipose tissue. So our objective is to establish the kinetic of visceral adipose tissue remodeling and dysfunction in obesity, and also to explore the mechanisms leading to cardiometabolic abnormalities. So in order to explore the early sequence of structural and functional and molecular abnormalities, we used the mouse model of high-fat diet-induced obesity because it recapitulates quite well the human obese phenotype. So we used 2-month-old mice and we fed them with either show diet or high-fat diet for 2 weeks or 5 weeks or 10 weeks. And we assessed global parameters such as body weight and cardiac function and metabolic function. And we assessed also local parameters such as tissue structure and function and molecular aspects, both in adipose tissue and heart, but also in adipose tissue explants and plasma. Our data showed that 2 weeks of high-fat diet were sufficient to promote senescence in visceral adipose tissue, and that this senescence was mostly localized within macrophages. So in order to confirm that adipose tissue macrophage senescence is indeed induced by high-fat diet, we did in vitro experiments. So we used bone marrow-derived macrophages, and we treated them with palmitate and high glucose for 24 hours. And we assessed senescence markers. So the senescence markers were indeed upregulated in response to this treatment. To further confirm the role of macrophages, we treated mice with clodronate containing liposomes in order to remove the macrophages, together with a high-fat diet for 2 weeks. And the data obtained showed that macrophage removal completely prevented metabolic alterations and cardiac alterations. So to conclude, this data that I showed and those that I didn't show here clearly demonstrate that metabolic alterations and cardiac dysfunction occur at a very early stage during obesity. Also that visceral adipose tissue senescence is a primary event and precedes inflammation and tissue remodeling. And especially that adipose tissue senescent macrophages are key factors in the early cardiometabolic abnormalities. So our aim now is to characterize senescent adipose tissue macrophages and understand the mechanisms leading to metabolic and cardiac alterations during obesity, and also to focus on the role of the secreted factors, also known as SASP or senescence-associated secretory phenotype, in the crosstalk between adipose tissue and heart, and identify potential therapeutic targets. Thank you for your attention.
So I hope that with this presentation, you are convinced that the adipose tissue plays a key role in the premature aging through cellular senescence of the adipocytes, but also of immune cells. And it's established also the importance of the crosstalk between different organs. So this is really an integrated process that probably opens the way to new therapeutic options and new prevention in those patients with obesity who are at risk for this premature aging and these comorbidities. I hope that you are convinced, Robert.
Yes, I am. Yes, I am. Yeah.
So we have seen the important role of the adipose tissue. We know that it is a buffer in normal conditions. And we understand that this is an important point in the current environment to be exposed to pollution. So what is the role of the adipose tissue in pollution?
Thank you, Geneviève. Yes, let me tell you more about this. This is not a very classical or usual function of the adipose tissue, but we know more about this lately. So to the left, you can see the maps, and in red, you see countries where the percent of obese individuals or individuals with obesity is higher than a certain limit. And in the upper part, this was in 1990, and in the lower part, this was in 2010. And so in a matter of 20 or 25 years, you see a huge increase in obesity in many, many countries, actually. So we have to now learn more about the role of adipose tissue. In the upper right part of the slide, you can see the adipose tissue with its functions. We know that it has a physical function. We know, obviously, that it has a metabolic function, an endocrine function, for example, with leptins and adipokines. And the question is, and what I would like to show you today is, does it have a toxicological function? And in fact, the interaction between toxicant, which is shown below, and a human being is in both directions. There is the effect of the pollution on human beings and the impact of human beings on the uptake and elimination of pollutants. And we will discuss both. Here we will discuss the role of pollutants on obesity and the adipose tissue. Several pollutants, mostly endocrine disruptors, are called obesogens. Why? This is because when, for example, organisms are exposed to such obesogen, and you can see in the figure of the mouse to the left, the mouse that has not been exposed to DES, which is one of these obesogen, and to the right, the mouse that has been exposed to DES early in its life. And you can see the impact, and you see there's the higher the obesity of the mice that has been exposed. All these compounds are called obesogens, and therefore there are a number of compounds. If they are added to organism early on in the life, they can induce an increased body weight later in life. This is shown in the diagram in the upper right part, where we show that there is a period of the lifetime, mostly the fetal life and the young childhood, where there is certain vulnerability of the individual. If there is exposure there to obesogen, then this increases the risk of having obesity later in life. How is this done? The most likely mechanism is epigenetics. And epigenetics, there are many ways of many mechanisms for epigenetics, including DNA methylation and histone modification. But basically what epigenetics does is not to modify the sequence of the DNA, but rather to modify the context and some of the chemical property of DNA and the proteins around the DNA and some messenger RNA. But these modifications can be stable and can be passed from one cell to the other. And in the lower left part, you can see an example of differences in methylation of a gene induced by a pollutant. So in the first, we have seen the effect of pollutants on the adipose tissue and how they can induce obesity through epigenetics. Now we will see that the adipose tissue itself can have an effect on pollutants, and we have many arguments in favor of that. First, if you are exposed to certain pollutants called persistent organic pollutants, and the good example is dioxin or some pesticides or PCBs, then these pollutants are actually stored in the adipose tissue. This is the first part that I call protection. Why do I call it protection? Because in fact, we know that this storage effect of adipose tissue actually protects other tissues of the organism from the impact of these pollutants. So instead of going to the brain, for example, these pollutants are stored in the adipose tissue. And you have a formula below, which is a little bit difficult, but basically what it says, in this formula, we have compared the impact of dioxin on a number of aquatic animals having different adipose tissue weight in these animals. And the more the animal had adipose tissue, the more they were protected towards dioxin impact. So this is evidence that probably adipose tissue can be protective. This is the first part. The second part, which is dangerous, is that since adipose tissue stores these pollutants, in fact, actually it can release them at a lower rate, but at a chronic rate. And this may be dangerous to the rest of the organism. So you are protected first, and then on a chronic basis, you can release these pollutants in the body and induce some negative effect. Is this right? This is seen actually in a study we have, in a clinical study we have done, shown to the right, where we have studied persons with obesity, with morbid obesity, undergoing stomach surgery and leading to the loss of several tens of kilograms in a few months. So these people lose a lot of adipose tissue. And what we show below is that when they lose adipose tissue, there is an increase in the amount of some pollutants in the blood. So they have released these pollutants in the blood. When you lose adipose tissue, you release pollutants in the blood. And this, we have also shown that it has some effects on, for example, liver biology. So to conclude, we can see that there is a strong interaction between adipose tissue and the pollutants, and particularly POPs. These are called, to the left, toxicodynamics of these pollutants. Basically, what these pollutants do on the body, on the adipose tissue. Many of them seem to increase the amount of adipose tissue, leading to obesity, probably through an epigenetic mechanism. Others have an effect on the growth of adipose tissue, its metabolism, and its inflammatory properties. This is on the one hand. On the other hand, the adipose tissue can store these pollutants. That's protective. However, under certain conditions, it can also release these pollutants on a slow rate, in a chronic basis, and this may lead to toxicity of the rest of the organism. So this is basically what I wanted to tell you about, the role of adipose tissue in pollutants.
So thank you very much, Robert. This organ, the adipose tissue, is really key, and you illustrated that in our current environment, it's also one of the targets of a pollutant. I had one question. As some study, you referred to dioxin, for instance, and in epidemiologic studies, a few of them reported an increased rate of metabolic disorders such as obesity, but also of diabetes mellitus in places which are unfortunately very well known with a very high rate of dioxin. So could you comment on this? Is this something which is confirmed? Is there a risk of living in certain areas which induces specifically some metabolic disorders?
Yeah, this is very important. This reflects the obesogenic properties of dioxin and many other compounds. And actually, this has been shown in several epidemiologic studies. However, I would like to take advantage of your question to make a word of caution in terms of it, because in those difficult areas, the effect is not only of pollutants on obesity. Obesity is a multi-parametric disease. There are many determinants of obesity. If there is an unbalanced diet, this obviously can lead to obesity. If there is not enough exercise, a lot of sedentarity, this also can lead to obesity. There are also some hormonal effects, some genetic effects. So we know a lot of determinants of obesity. What we can say is that pollutants are one of these determinants, but it's at this stage quite difficult to tell you what % of obesity is related to pollutants. This is a more difficult question that we are studying currently.
Thank you, Robert. And I think that the study of the cohorts and the follow-up of this cohort together with an environment and the clear definition of the pollutants will be very important to better understand this question. This comes to the point that I would like to ask to Isabelle, because we are speaking about pollutants as a sort of generic words, but it's probably more complex. So Isabelle, can you explain us what are the main pollutants, what should be aware as biologists or physicians about the risk of specific pollutants? Can you?
Well, as far as the atmosphere and the air quality is concerned, most of the interest is focused on regulated pollutants with proven health effects, such as fine particles or nitrogen dioxide. But they are, of course, only traces of pollution, which contains a very large number of species. In cities, this pollution is very heterogeneous, and it is particularly strong near roadways due to vehicles emissions, but it is also strongly expressed in homes or inside the vehicles. So as it depends on where we live and how we live, there are very strong inequalities in exposure to pollution in urban areas. In fact, epidemiological studies have been able to highlight the phenomenon of exposure contrast, which is the deterioration of the health status of populations living near high traffic areas. So to characterize this exposure and address the question of its impact on health, it is thus necessary to develop fairly advanced measurement or modeling tools. So we asked Arthur Elisa Etuman, a postdoctoral student in atmospheric science with strong skills in interdisciplinary approaches, to present his research work with the aim of providing consistent decision support for environmental public policies. He indeed has been developing a fully integrated model to connect the urban organization, people activity and mobility, the related energy consumption, and the calculation of air quality and exposure. So such approaches allow to better understand the determinants of exposure to pollution and the societal and research issues that are associated with them.
Thanks for having me on EUR-Live. I'm Arthur Elisa Etuman, postdoctoral researcher at LISA and at University of Paris Sécurité. I will talk about scientific and societal challenge of air quality. Urban air pollution is one of the main challenges of our current society, simply because on one hand, more and more of us live in city, and on the other hand, we live close to the main source of urban pollutant. Nowadays, the fight against urban air pollution is correlated with certain issues related to environmental policies to combat climate change, but also to urban planning and local transport plan. And more than ever, it's necessary to predict and quantify the potential impact of this plan and to understand in a global vision. How can we change our daily mobility? How can we reduce the share of fossil fuel? How can you reduce the exposure of citizens and highlight social injustice in terms of air quality? And it's in this purpose that I'm doing my research on numerical modeling on air pollution. I'm currently working with several scientists from different laboratories, such as LVMT, CERED, to model the impact of urban planning on air quality as part of the project Polexpo, which is funded by the National Environmental Agency of France. The modeling platform, as we can see here, starts with the transcription of urban policies into land use data. Here we try to recreate the dynamic of the urban fabrics, such as a place of work, residential area, and we also consider a warehouse for the good transportation. This modeling of urban dynamic allows us to produce emission data of pollutant by considering key sectors such as road transport, but also household energy demand. Of course, all the sources are considered such as biogenic and industrial emissions too, and in additional, meteorological model are integrated, and also potential pollution from outside of the domain are taken into account with the boundary conditions. Population exposure is achieved by cross-referencing air quality data with population data. To integrate the effect of urban policies and energy consumption on air quality, we have focused our approach on the Olympus modeling platform, which is a multi-agent system platform coupling 6 different types of models. The first model generates an artificial population based on Bayesian statistic and census data. The second model generates the activity of the agent, which are predicted on the basis of a gravity model. The timetable and the travel demand are realized with the activity travel demand model. And for the trip by car, the demand is affected and assigned on the road network based on the graph theory. At the same time, the energy demand model fed by the artificial population model give us the energy demand per household. Finally, the model produces the emission from the road transport and the residential sector, which will feed the air quality model. In the case of the Île-de-France region, we were able to produce some scenario based on the 2030 master plan of the region. We have been able to predict the evolution of the land use, as we can see on this figure, but also the evolution of the construction according to housing and transport policies. We were also able to model artificial population, as I showed you before, and its emission condition by future transport infrastructure that will affect the choice of agent in their mobility. On these last 3 figures, we try to highlight the difference between a business as usual scenario and a densification of public transport network in terms of emissions, concentration, but also exposure of the population. Thank you.
So this work presented by Arthur shows us at the same time the very great complexity that lies behind the estimation of exposure to pollutant for individuals in a city, as well as the difficult task to provide decision support for policy. And this plays a very strong role in defining what will be the health trajectories of urban population. But it makes me think that it's not only air pollution that impacts our organism, then it should not be the only thing taken into account. So this ambient pollution and ambient exposure leads us today to ask ourselves the much broader question of our total environmental exposure in the form of what is called the exposome. So Robert, maybe you could talk to us about this concept.
Yes, let me tell you a few words about the exposome, and let me try to convince you that it is a useful concept. Next slide, please. First, I think we have to show how important pollution is in general. This is a slide coming from WHO, the World Health Organization. So this is serious stuff about the number of deaths depending on different factors. And you can see different factors such as exposure to pollution, to tobacco smoking, to several infectious diseases. And in this study of WHO, you can see that pollution is actually quantitatively the most important factor leading to death in the world. So this is something important we have to address. But all the others are important too in terms of exposure. So next slide, please, will show you what is this exposome concept. In fact, it was Christopher Wilde in 2005, like 15, 16 years ago, so that defined the concept. So the concept is still in its puberty, we can say. And why did he feel like defining such a concept? In fact, he was working on cancer, and he knew a lot about the effect of genome on cancer. But he was also interested by all the other factors, and it was a little bit of sort of a mess with the other non-genomic factors. So he coined the term exposome as the life course environmental exposures, all kinds of exposures, chemical, biological, physical, psychological, all kinds of exposure together, all during all the life, from prenatal to all the way to death. So this is really something extremely large, and actually in the US, some people call it the wild concept. But I will show you that it is useful, and some other authors have tried to focus a little bit more on some aspects of the exposome. For example, Rappaport focused on the chemical exposome, stating that by analyzing, for example, blood, human blood, you can learn a lot about the exposures and some of the other chemicals that we have in our blood. Miller, who is a toxicologist, actually linked exposure to effects. As a toxicologist, he was also interested in effects, and for him, exposome covered both the exposure but also their effects. And there were several reports defining the eco-exposome, where the focus was not only on the human population, but also on the ecosystems and the relationship between the ecosystems and basically the human population. So what does it encompass, the exposome? It is actually, I have to say, quite complex. However, we can do something to analyze it. Why is it complex? There are several stressors, as I stated: chemical, physical, biological, psychosocial. What is important is the source. It can be from air, as we have just seen a little bit earlier, but also from water, from soil, from food, as the other example we had from the student, but also consumer products and drugs and tobacco, etc. Where it's quite important, whether it's at work, at home, in the metro. We also, when is very important, because you do not have the same effect if you are exposed, for example, to a chemical, if it's during the fetal stage, the childhood, or at the adults. So very important vulnerability effect. And also what type of effect, which organs, which tissues, where does it go? So there are a number of issues that we have to study when we're studying the exposome. So is it feasible? How can we explore all this? Let me give you an example from a European study that we contributed to. It's called HEALS, and it tried to explore all these exposure. First, there are many tools that you can use. For example, in the upper left, you can use satellite to inform about, for example, the weather, or how the greenness, how much trees you have in a city, or how much water you have, etc., etc. You can have a lot of information from satellite, but you also can have a lot of information from sensors that you can put in the environment. For example, how many particles do we have in air, or on yourself, and this can measure, let's say, noise, also particle dusts, or many chemicals. So you can learn a lot with these sensors. There's a lot of activity on this. To the right, you have modeling. Modeling is extremely important. You cannot see much here, but basically what it says is that by knowing more about the contaminants in air, in water, in food, you can probably use a model to deduce how much exposure human population are undergoing. So this is part of the modeling systems that we use a lot in exposome studies, very helpful. Next, please. Then you can see in the lower left that you can do a lot of studies on biological matrices from human, for example, from blood, urine, or sometimes hair. You can analyze a number of chemicals, and this number is really growing, but you can also analyze effects. For example, using omics like transcriptomics or proteomics, a lot of information coming here, or some other physiopathological data. So this can be done, for example, using in epidemiological studies, called EWAS, where you try to put a relationship between environmental exposure and some type of health outcome. For example, we have seen earlier obesity. You can do this kind of study. And also you can use toxicological study, molecular studies, to establish causality, because most of the time epidemiology leads to association. However, toxicology, experimental work, and modeling can support the causality between an environmental insult and an outcome, a health outcome. So there are a number of implications for these exposome studies. They are feasible. First, this has triggered really a considerable innovation in tools that we can use and in different disciplines. For example, sensors have developed considerably and can be used and will be used a lot. As I said, monitoring, assaying in blood, for example, how much exposure to chemicals do we have, or assaying how much noise there is in the environment. Modeling is extremely important. We have seen exposure modeling we can see, the link between environment and health. And there's also modeling inside the body to know, for example, if I assay a chemical in blood, how much there is in brain, for example. We can also study effect markers, therefore impact of the exposures, and we can assess combination of effects. And that's a very, very relevant issue. And we call it systems medicine, where you have different information and you put them all together to try to deduce some, for example, diagnostics. It has supported, the exposome has supported the multifactorial concept of disease. We have taken already the example of obesity, and I showed you that obesity is related to some pollutant, but also to diet, also to behavior such as physical activity or sedentarity, and many other outcomes. So this has to be studied in an exposome study, obviously. It promotes multidisciplinarity. You need mathematicians, people who analytical chemists, you need epidemiologists, toxicologists, a wide range of people involved. And it has implication in public health. For example, we know that we should study a number of chemicals at the same time. And in fact, currently the regulation is done each chemical by itself, as if nothing else existed. And this is probably not enough. And the exposome will probably be helpful in this respect. It has also focused on vulnerable population, and this is quite important in public health. And I think it will influence public health messages, the coherence of public health messages. At a more global, at the individual level, I think it will trigger something called precision prevention. You know how prevention is important, but the more we know about the exposures and the different factors, the more we know quantitatively about them, how to assay these, how to know more about the chemicals we are exposed to, or noise or radiation or whatever, the more we can be precise in our prevention. For example, somebody who has asthma knows that if there are some chemicals around, some particles around, or ozone around in the air, he has to be quite careful. This is one example. Or somebody that has diabetes would like also to know what kind of environment is around. So these are some of the implications. We hope that with this concept, which is extremely wide, in fact, can be useful at the end of the day.
Thank you. This is a fascinating research area. And you're right, the synergy effect still needs to be addressed. I was wondering today, how many chemicals are we able to detect in the biological analysis?
Yeah, this was one of the major development in the technology. I mean, initially people use a lot of NMR to detect chemicals, but lately we have used much more something called mass spectrometry. If you have went through a chemical curriculum, you know about mass spectrometry. And mass spectrometry, for example, in my clinical lab, biochemistry clinical lab, we use it quite a lot, and we assay for like 200 chemicals. But now we have much more sophisticated and high resolution mass spectrometry that can give an idea about thousands of chemicals. One of my friends, Miller, he says when you do genomics, the gene level, you have sometimes 1 million of variants, and we only deal with a few hundreds of chemicals. So the technology has developed, and I guess in the future we will be able to assay thousands, maybe tens of thousands of chemicals, and try to see what this can do.
Thank you, Robert. That is so fascinating. You have spoken in depth on chemical and these chemical aspects. We understand now that being obese and living in an atmosphere with many pollutants is a double burden, and you may have some additional data to provide. What about the noise, if you add this on the top of all these aspects on pollution?
Very, very interesting. Yeah, first, yes, the exposome studies are expected not to focus on a single stressor, or a single chemical, or even a few chemicals. It's really supposed to show the interaction between different types of stressor. Let me give you an example. We have studied the effect of high-fat diet. We know that high-fat diet, it may be nice to eat high-fat diet, but in fact, for our health, it's really not so good. We have studied the interaction between high-fat diet and dioxin, which is a pollutant. And we have seen when we combine both in experimental studies in rodent, the liver underwent a change in its histology that is reminiscent of a human disease called NASH, steatohepatitis, non-alcoholic steatohepatitis, where there is fat in the liver, where there is fibrosis in the liver, and where there is inflammation in the liver. High-fat diet alone does not lead to that. Dioxin alone does not lead to that. When you put both together, then you have this effect. And you mentioned noise, physical stressors. These are maybe, except of radioactivity, these are maybe the least studied stressor. And in fact, epidemiologically and clinically, they are very important. We know how much noise is important, not only for audition, but also for lack of sleep and for cardiovascular disease. There is evidence that it has a role on cardiovascular disease. So it has now to be studied. We can use sensors for that, that can assess quantitatively noise and make some very good exposure studies of noise. This is now being done currently.
Many thanks, Robert, and many thanks, Isabelle. We have now reached the end of our program, and just to summarize for the students who would be interested in joining this program of aging and vulnerability together with the impact on environmental factors. You have here a graph, a cartoon summarizing how we envision the link between cell senescence and pathological aging under the influences of genetics, but also environmental factors, including not only pollution, but also nutritional factors. And we have a program which is interested in deciphering the link between the cell senescence and the organ damage and how it impairs and induces chronic diseases and how it impairs the prognosis of these chronic diseases. Next, the program that we propose is quite large because from these cartoons, you can see that we are coming from very initial studies at the basic scientist level with preclinical studies in rodents. We can move to clinical trials because we have cohorts of patients that needs the skills that we are combining between our different institutions and labs. And we hope with this to provide interesting work and new products to tackle the problem of aging in the current environmental factors. So those who are interested in joining this program, which was an introduction today, you are very welcome to have a look at this program, which will be present in the website of EUR-Live. And we have next course for master and PhD students in June and May. So do not hesitate to apply, and we'll be happy to work with you. Thank you very much. Thank you, Robert. Thank you, Isabelle, for sharing with us this fantastic program, and hope that it will be that it will encourage the students to join us. Thank you.
Thank you.
Thank you.